I'm going to talk today about our invention of virus-like particle vaccines for opioid drugs. My name is Kathryn Frietze. I'm an assistant professor in the Department of Molecular Genetics and Microbiology, and I'm also a KL2 scholar in the Clinical and Translational Science Center here at UNM. So the work that I'm going to talk to you about today includes the following inventors: myself, Bryce Shakarian, who you saw a picture of earlier as a innovation fellow, Susan Kaur, who is a senior research specialist in my lab, and Dr. Naomi Lee, who is at Northern Arizona University and is our collaborator. So far, we have received some funding for our work. We received an NIH administrative supplement several years ago to do some initial work, which I will share with you today, showing proof of concept for using virus-like particles for opioid vaccine development. And then I am supported by the UNM Clinical and Translational Science Center KL2 Scholar Award, so that is also funding that has gone toward this. So our invention is using bacteriophage virus-like particles as an immunogenic platform for opioid vaccines. And so basically the idea is we have these bacteriophage virus-like particles that we have been using for other purposes, primarily for targeting cancer or targeting infectious disease. But we thought that it would be also a good platform to use to target opioids. And so what we do is we have a bacteriophage virus-like particle shown here in blue with the yellow dots. And what's unique about these virus-like, sorry about that. What's unique about these virus-like particles that we use is that they have these surface exposed lysines that are able to be used as a way to connect things onto the surface. And so we use what's called a bifunctional crosslinker, which is this chemical shown here called SMPH, which allows us to connect a molecule, in this case, opioid drugs of interest, to the surface of these virus-like particles. And we do this by modifying the opioid drug itself to include a short linker sequence that includes a cysteine amino acid, which provides a functional group to be able to attach the drug to the surface of the virus-like particle. And so you may be asking yourself how opioid vaccines are supposed to work. So we all know there's an opioid epidemic going on, individuals who are suffering from opioid use disorder or addiction, and also a lot of people dying from opioid overdose. And so there's this idea that if we can elicit high titer antibodies, which are molecules that float around in our body and usually target infectious diseases, if we can trick our immune system into making these antibodies to the opioid drugs, then when an individual is exposed to the opioid drug, whether through intravenous injection or ingestion or inhalation, then those antibodies will bind up to the drug and prevent the drug from entering into the brain where it acts. And so this has been actually proposed as a potential treatment for opioid use disorder and also potentially for opioid overdose. Currently, opioid vaccines that are in development require multiple immunizations to be able to reach the peak titers necessary to protect against the opioid drug. And even when these opioid vaccine strategies do elicit antibodies that can work, these antibodies quickly wane in the body. And so it requires repeated immunizations to be able to reach enough antibody to be able to protect somebody. And so our approach is different in that we're using a really highly immunogenic platform, the bacteriophage virus-like particles, to be able to elicit these high titer antibodies against the opioids that hopefully will last a long time, and also that you will quickly get antibody responses. So Going along with that, as I mentioned, we all know the opioid epidemic is a really big problem. This graph here from the CDC is just showing sort of the extent of the problem that, over the past approximately 10 years, we've really seen a huge increase in the number of overdose deaths caused by opioids. And that's really being driven by synthetic opioids, primarily fentanyl, and both fentanyl that individuals are taking for medical purposes, fentanyl that is added into street drugs to make them more effective for the individuals who take them. So some people are overdosing on accident, not knowing that they have been exposed to fentanyl. And so our idea with our vaccine is that if we can, in fact, elicit really high titer, long-lasting antibodies against opioids, then we might actually have a chance to block opioid overdose in susceptible people. That's important because no opioid vaccine so far has shown to actually protect against lethal overdose, and we think our vaccine has a chance to do that. And also no opioid vaccine, as I mentioned, is able to reach acceptable titers within a single dose of the vaccine. And we think ours might be able to do that. Just a little bit more information about the scope of the problem. So worldwide, about 16 million people have had or currently suffer from opioid use disorder. Treatments are really limited. Medication-assisted treatment, which includes methadone, buprenorphine, and Suboxone, is really difficult to access. There's a lot of federal regulations surrounding providing these drugs to patients. And so even if individuals are able to access these drugs, it can be a huge barrier to being able to get regular treatment. In 2019, 50,000 people died from opioid-involved overdose. The best strategy we have right now for helping with the opioid overdose issue is naloxone or Narcan, which can actually reverse an opioid overdose. However, it requires rapid administration. So you need a person who has this drug available on them to give to the person who's experiencing an overdose very quickly. And so in both of these situations, having something like an opioid vaccine that can provide an additional treatment option or prevention option for opioid use disorder or opioid overdose would be really important. I also just wanted to mention that there is a national initiative that was started by the NIH called the HEAL Initiative. It stands for Helping End Addiction Long-Term. This is providing funds and also assistance from the federal government, primarily the NIH, to develop new treatments for opioid use disorder and opioid overdose. And the funding that we've already received has been from this HEAL Initiative. And just also to mention, one of the focus, the research focus that HEAL has is to help develop opioid vaccines. So there's a lot of interest in the country for developing vaccines like ours. I just wanted to show a little bit of data. So I mentioned that we really think that our vaccine has an opportunity to prevent overdose and also can help individuals who are experience or suffer from opioid use disorder. So the first thing that we did is we generated our bacteriophage virus-like particles displaying these modified opioid drugs. And so we've shown so far that we can do this with three different types of opioids. So in this first graph here, I'm showing Q-beta oxy. So this is oxycodone attached to the surface of the Q-beta virus-like particle. And then in this other picture, we have both morphine and a molecule called 6-acetylmorphine. Importantly, morphine and 6-acetylmorphine are the active metabolites of heroin, which is a major target that we're interested in pursuing. The graph that I'm showing here is the antibody titers in mice after we immunize with a single dose. So we take a group of mice, we immunized with our oxycodone Q beta vaccine or just Q beta alone. And then we looked at how quickly the antibody response comes up. So we took blood samples at day 3, 7, 14, 21, and then on to look at how long the antibodies lasted as well. And so over here in this graph on this x-axis here, we have days post-immunization. And then on the y-axis, we have the actual antibody titer. And so as you can see, as early as 3 days post a single immunization, we start to see antibodies appear in the mice. Those reach high titers by about 7 days, and we reach peak titers around 14 to 21 days. And as far out as we've seen so far here, this is 112 days post-immunization, we still have high titer antibodies. The other lines down here, I especially just want to draw your attention to the black triangles. This is oxycodone conjugated to a protein called KLH, which is a competitor strategy. And you can see that with a single immunization, there's much lower peak titers that are reached, and these wane very quickly. So we think our strategy is going to be really a lot better than competitor strategies. So just the current status of what we're doing. We, so far, as I showed, we've made these 3 different opioid targets: oxycodone, morphine, and 6-acetylmorphine. We've done that same experiment that I just described, and we see the same response in all of them. So all of those different targets, we can elicit really high titer responses very quickly that last quite a while. And so the next steps that we're focused on, one is we're looking at binding specificity using just a single dose or 2 doses of vaccine. This is important because as we move toward thinking about putting this into patients, it's going to be important to make sure that the antibodies that we elicit are binding to the drug that we want and not other things like endogenous opioids or other opioid drugs that we might not want to target. We're also going to be assessing lethal challenge after 2 doses. We did some preliminary experiments that show we may be having some effect with lethal challenge with just a single dose very early on, but we want to expand these studies. We're also interested in doing mouse studies to look and see if the vaccine is able to block drug activity. And then also we have experiments that we're hoping to do in non-human primates, rhesus macaques, where we look at the kinetics of the antibody response as well to see if they have these quickly rising antibody titers that last a long time. We have some funding pending. So I just submitted a School of Medicine RAC pilot award to do those mouse challenge studies, both lethal challenge and also looking at drug activity. We have submitted a UG3 UH3 NIH grant that would develop, use the same strategy for developing vaccines against fentanyl and heroin. That is close to $3 million direct over 5 years. And we're waiting to hear if we receive that funding. We also applied for a pilot award in collaboration with the California National Primate Research Center to look at the kinetics of the antibody response in non-human primates, and we should be hearing about that soon. And in terms of collaborations that we've established, we have a collaboration with Bruce Blow, who's at RTI, Research Triangle International, and he's a medicinal chemist who can make some of these compounds. Herman Statz at Duke University, who's an immunologist, and then here at UNM in the pharmacy department or School of Pharmacy is Matthew Campen, and we're planning to do some different exposures and test our vaccines with him. Finally, I just wanted to give an additional acknowledgement to my laboratory. Everybody has sort of been involved in this this work in some ways, but I wanted to highlight especially Isabella Murphy, who's a new graduate student in the lab who will be taking on this project for her, or part of it for her graduate work. And then Enzoa Demuz, who is another graduate student in the lab who worked on some of this for her rotation project. And I will end there and I can answer any questions.